Very good afternoon to everyone. In the next 20 or 25 minutes, I'm going to share with you our experience with the drought in maize. Before going into the details of the experiments, I would like to share some statistics. The area of cultivation of maize in India is about 8.5 million hectares, and the production is about 22 million hectares, with a productivity of 2.54 tons per hectare, which is far less than China and USA put together. The half of the maize produce is consumed by poultry. There is an estimation that the demand for maize may go up to 45 million tons in India in the coming years, owing to ever-growing needs from various sectors. For example, the purchasing power of people in India is increasing. They are affordable to buy meat and meat products. So the poultry industry is getting expanded. So more maize is needed to feed the poultry industry. So how to improve the productivity? So I told that the productivity of the maize in India is just half of the productivity of China and USA. Tremendous opportunity is available to improve the productivity. The one way is to breed better cultivars, and the second best option is to have some better management practices. Sometimes despite having good cultivars and management practices, the productivity is affected by biotic and abiotic stresses. Among many abiotic stresses, I would consider drought is an important issue not only for India but also globally. This is the IPCC weather report. Almost, all continents, most of the world is going to face the drought-like situation in the next couple of decades. So we have to be ready to face the challenges. In India, 70% of the maize is sown during Kharif season. It means June, July. Depends upon the commencement of the monsoon. The failure or inadequate monsoon will create drought stress, and it may occur at any point of the crop growth stage, starting from seedling to seed setting. Particularly drought at flowering and seed setting stage will reduce the yield significantly. For example, drought at flowering stage will cause pollen sterility or abortion, whereas drought at seed setting stage will affect the yield contributing traits such as number of grains, grain yield, and weight, and so on. So at IARI, we have started series of experiments to understand the functional mechanism of drought tolerance and to breed better cultivars. The objective of our first experiment was to genome-wide assembled nearly 264 genotypes collected from different breeding centers of India, and these genotypes were grown at 3 locations: New Delhi, Hyderabad, Karimnagar, for 2 consecutive years. Stress was initiated at the time of flowering stage, and several traits were phenotyped, including anthocycline interval, grain yield, and yield contributing traits. The same panel was simultaneously phenotyped by using 56,000 SNP array, and we have generated nearly 13.4 million SNPs across the panel. The coverage of SNP across the genome was about 2,200 for chromosome 2 to nearly 5,500 for chromosome 1. This is a linkage disequilibrium map where you can see the high and low LD blocks. And these are all the sum of the high LD blocks on chromosome 3 as well as on chromosome 8. We have also measured the LD decay, and the mean rate of LD decay in our population was about 200 to 300 kb if you consider R square at 0.1 level, whereas it was 5 to 10 kb at R square 0.2 level. This kind of low LD was expected in our population because most of the lines in our panel was consisting breeding lines, so we assumed that some of the loci were already fixed in the population. This is the per se LD pattern of the genome, and where you can see some of the high LD regions at the centromeric region and low LD towards the telomeric region, which means that the active regions of recombination present towards the telomeric region. We have also worked out the population structure, and In fact, that the panel showed some kind of structure. The number of subset in our panel was 4, which was fixed by the cross-validation error from admixture model. And simultaneously, we have worked out the physical components, and the grouping pattern of the principal components was closely matching with the grouping pattern of the admixture model. Then the data was analyzed using different statistics. The first model in Genable includes the basic data plus inputs from multidimensional scaling plus FTR correction. The second model includes inputs from population structure in addition to the existing variables, whereas in GATIT it uses principal components in place of this MDS. We have come across some interesting results while correcting the population structure. For example, if you consider only the basic model K for the anthesis silking interval data from Hyderabad, the number of false positives were very high, but the number of false positives were reduced and eventually reduced by adding additional models such as PCA or structure to the model. So these are all the significant SNPs we have identified for anthesis silking interval across data. This is the comprehensive p-value heat map of all significant SNPs we have identified across data. I told that we have used 4 models, and 3 location data, and SNPs were detected across 7 tribes. A total of 67 significant associations were detected across data, of which 17 were FTR corrected. Anthesis silking interval has more number of associations followed by grain yield. Apart from this significance and false discovery rate correction, we have also looked into the consistency of the association across correlated tribes. For example, the SNPs mapped on chromosome 5 for grain yield have also been mapped for yield contributing tribes. So this kind of consistency actually were increasing the reliability of those associations. So the question is, we have got significant SNPs, whether these SNPs are associated with some kind of stress tolerance. So this is the grain yield data that look at this locus that was consistently detected across model and locations. And this is the zoom in view of the locus. Here we have searched the presence of genes in the vicinity of the locus using maize gene models. And the results were very much encouraging. We have identified several genes, and some of them were really associated with stress tolerance. For example, MIB and ethylene responsive factors and binding protein, these are the elements we have identified in the vicinity of this locus. So in the same manner, we have identified genes either directly or indirectly associated with stress tolerance from all our significant SNPs. And out of 67 significant SNPs, 22 were associated with the stress tolerance. So this is the collective, we are proposing the collective role of SNPs identified across the data. The outer circle shows the number of the genes identified from our experiment, and the second circle represent the possible molecular mechanisms, and the third circle is the traits that are controlled by these genes. In fact, that we have identified several important genes related to stress tolerance, such as GIT, MIB, NASB, and so on. So the point is, we assume that these set of genes should have been played a major role in maintaining the biological functions during stress conditions. As a follow-up, as well as to validate the results from the GWAS experiment, we have collected tissue samples from them on pathways like photosynthesis, carbohydrate metabolism, signaling, and so on. But we are more interested to see the interaction of these genes in different biological pathways. For that, we have collected a huge volume of data, nearly 544 microarray samples from public domain, and we have created these co-expression maps, and we have mapped our expressed genes. You can see the clear-cut difference between between these two genotypes with reference to their mapping structure. And to reduce the complexity of the map, we further split the map into different modules. The number of modules for tolerant genotype was 10, whereas it was 8 for the sensitive one. Apart from that, we have identified 174 drought-related genes, and we have also mapped these 174 genes onto the modules. The interesting point is, out of these 174, 75 were unique to the tolerant genotype, and these transcriptomes were not at all expressed in the sensitive one. Apart from that, genes such as heat shock proteins was co-expressed with 19 other DEGs. This MYD was co-expressed with 50 other DEGs, and this ERF, ethylene responsive factor, was co-expressed with 84 other DEGs in the tolerant map. These kind of extensive interactions were totally missing in the sensitive one, which means that the tolerant genotype has different kind of expression and was showing a greater interaction with other genes than the sensitive one. I told that we have identified 174 stress-related genes, and we want to see the interaction pattern of these stress-related genes. For that, again, we have created a specific co-expression map using these 174 genes, and they form nearly 11 clusters, and each color represents one cluster. In cluster 1, the genes are involved or related to stomata closure. These are the genes in cluster 1. And this is cluster 2, where here they include genes related to signaling and protein cascade. And the cluster 3 includes genes related to photosynthetic activity. Here we have seen that the genes that are expressed, that are having similar function, they group together. And the second point is stress-related genes in different clusters were extensively interacting with each other. And the third interpretation is, among different clusters, 11 clusters, genes involved in stomata closure, signaling, and photosynthetic activity were highly interacted with other clusters. So this is the outcome of the transcriptome assay. The hypothesis is, in fact, the drought triggered series of transcription factors and genes, and those genes got activated and in turn regulate different metabolic activities like carbohydrate metabolism, protein, amino acids, raw scavenging, and so on. To cut this story short, the tolerant genotype is superior in maintaining the photosynthetic activity and other metabolic activities. And the tolerant genotype is better in osmotic adjustment as well as water balance as well as raw scavenging activity than that of the sensitive one. So this is the kind of outcome we have got from the transcriptome assay. So just for curiosity, we have extended our work to other species also to see how genes are expressed in other species when compared to the maize. For that, we have collected nearly 1,500 array samples from rice as well as 900 samples from wheat in addition to the maize samples. And we have created this co-expression network as well as module-wise co-expression networks. And you can see the clear-cut difference with reference to their pattern and structure. So it means that the genes are expressing differently in different species. Here we are particularly interested in some of the important transcription factors related to stomatal closure, osmotic adjustment, and water balance. Further, we have shortlisted 17 transcription factor families, which include this HLH, C2H2, NAC, ERF, and Wiki families. The point is also the number of families are same across these species. The maize has only 290 genes, which is 30 to 35% lesser than that of the other two monocots. This is probably showing the efficiency of C4 over C3. Apart from that, this is the co-expression map of the maize for these 17 families. The HLH, this transcription factor has highly connected with other families, then followed by C2H2, then by NASC, then by ERF, then by Wiki. Whereas in rice, the order is something different. HLH followed by C2H2, followed by ERF, then WIT, then NASC. Wheat has totally different order. Again, it means that though the genes are same, their interactions are totally different in different species. Finally, we also wanted to see the gene regulation at post-transcriptional level. For that, we have collected we have collected the microRNA from genome sequence databases as well as array databases, as well as we have identified the target mRNAs. And we have, from using different tools, we have identified 126 matured microRNAs. The question is whether these microRNAs are playing any role in our genotypes. So we have collected 42 interesting mRNAs from our transcriptome experiment, and we have measured the transcriptome level of this miRNA-mRNA pair in two contrasting genotypes, that were part of the transcriptome experiment. I would like to highlight one important result. These are the three miRNAs and their target mRNAs, and these are the 6 elements controlled by these mRNAs. Here the point is the miRNAs were down-regulated in the tolerant genotypes and up-regulated in the sensitive one, whereas the mRNAs were up-regulated in the tolerant genotype and down-regulated in the sensitive genotypes, which means that the miRNAs were negatively regulating the important genes in the susceptible genotypes, leading to susceptibility. So that kind of conclusion we have arrived. So in summary, we have identified genes and SNPs responsible for important biological functions, including photosynthetic activity, osmoregulations, raw scavenging activity, and other metabolic processes. Right now at IARI, we have identified founder lines related to having some stress tolerance, and now we are combining the alleles to get better allele composition to breed tolerant cultivars. So my sincere acknowledgement goes to our donors and collaborators as well as research fellows and technicians. Without them, this work could not have been possible. Thank you very much.
Thank you very much, and thank you for keeping to time. We've got opportunity to take 1 or 2 questions. Roberto again.
Thank you for your nice presentation. I couldn't pass this opportunity. I work in maize and drought, so very interesting work. Unfortunately, I had to go out for 5 minutes, so I'm not sure if you talked about that or not, but I assume that the water balance of the plant came into the picture at some point, as you alluded at the end. So to what extent the different expression of genes that you find are just expression of different water levels? Because as a breeder, I think I'm more interested in the cause rather than in the symptoms of what I'm trying to breed for. So basically the point I'm trying to get to, did you have data also on the water status of the plants, and have you done a QTL analysis or some sort of analysis to identify which morphophysiological traits might have been linked to this different capacity of the plants to maintain water status? Roots, waxes, escape due to flowering time. If you could elaborate a little bit on that, I think I would appreciate it.
Yeah, I understand that. The point is, as far as this transcriptome assay is concerned, that we have not measured that so many physiological traits. It's only the outcomes are based on their functional annotations, right? Then it would be really nice, but we have got the 2 contrasting genotypes. They are really working, showing so contrasting and stress tolerance. Then it would be really nice to see. Even I personally interested to phenotype almost all physiological traits. Then we can even mapping those QDLs, and we can just validate those genes, whatever we identified from the transcriptome experiment. But right now, I don't have the physiological data related to those 2 contrasting genotypes.
Okay, thank you.
But we are planning to do that.
I think
yeah, we have developed mapping population, but just as part of the story, We are doing some QTL mapping experiments also
Again,
to validate the genes that are identified.
as a
from the-
breeder, I'm more interested in the cause rather than in the effect.
Yeah, I understand.
Although it's important to know what the symptoms are.
Yeah, my understanding is it's not a single trait. I mean, everyone knows it's a very complex, again, it's a quantitative traits. I really, I'm more interested in the interaction rather than the single trait based, approach. So what are the traits, that are combining together then providing the final result is more important to me rather than working only for the single trait. That kind of approach, we have been following here.
Thank you.
Question over here.
I don't know if I remember your point that you the last 3 genes, and I think you think you present that drought tolerant and drought sensitive, and why drought tolerant those 3 genes are downregulated and the drought sensitive are upregulated. Can you give an explanation?
Okay, you are talking about the last slide, the mRNA. Yeah, right. The point is, it's not only those 3 genes. Just I have highlighted only those 3 genes. We have got several set of genes actually. But the point is, for a trait, it is come from the DNA sequences to transcriptomes to protein, right? For the trait expression. Sensitive genotype doesn't possess those kind of traits which are blocked by those miRNAs in the middle. That is the idea. So you have the DNA sequence, the tolerant genotype has the transcriptome, and finally it is going to the some of the channels for final expression, whereas the sensitive genotypes, the expression is blocked in the middle of the gene regulation that was due by the miRNAs.
Okay, we have.
Yeah. Yeah, that is a it's a kind of the study. This kind of studies are evolving. The miRNA is also playing a crucial role in gene regulation, and most of them are actually negative. Yeah, of course, that is another study. They are also regulated. So mostly they are negatively regulating the things, regulating the transcriptomes. So sometimes they are destroying the transcriptomes during the process of gene regulation. So we have just highlighted those miRNAs, what we have identified. And these RNAs are related to some cis elements, and they are related to some kind of physiological mechanisms related to stress tolerance. Pardon me? Yeah, definitely. Yeah, it's going on.
I have a small question. With regard to microRNA mediated regulation of genes, did you see in a situation where single microRNA regulating more than one gene?
Yeah,
or
it is.
more
Yeah.
more than one microRNA regulating the same gene?
No, it's mostly, even if you scan the whole genome, you may not be able to get not more than 200 to 300 miRNA families, which means that one miRNA family is controlling or regulating more number of transcriptomes, right? So yeah, number of miRNAs at the whole genome level is very, very less when compared to the transcriptomes. Yeah, it means that one family is controlling more number of
More than one.
more than one gene or transcriptome.